welcome to the last chapter that we will do in our series of biochemistry videos. So this is based on the last major macromolecule we'll talk about. Also, so again, we started with carbohydrates, we went to lipids, we went to proteins, and now we're at nucleotides and nucleic acids. You might be familiar with these. Maybe you've heard of DNA and RNA before. So that's kind of what we're going to talk about. This is a relatively short chapter, so I'm not going to do a ton of overview. But generally speaking, we will talk about the structures of DNA and RNA, so what makes them distinct and different. And then we will talk a little bit about how we go from DNA from the parent to the offspring and just a general cursory overview of some of that you may have heard of before. But if not, great, then we'll have an introduction to it for this chapter. So, very quick, you may have heard of something called the central dogma, but the central dogma in biochemistry or in biology basically says we can take DNA, and DNA is used to make RNA, and then we can use RNA to make protein. So here's the difference, is that DNA and RNA, this is the same language, but slightly different. So again, this is the nucleotide language, but there are slight differences between DNA and RNA. So this is what's called transcription. So you're transcribing something. So imagine if you are transcribing something, you may be able to write everything down, but you may not get voice inflection or something if you're at a live lecture, things like this. Okay, so it's the same language, but it's just slightly different. And then finally going from RNA to protein, here you're going from a nucleotide language to an amino acid language. So because of that, you're actually changing languages. So this process here is translation. So you need a translator to do this. And the translators include something called transfer RNAs as well as the ribosome. So we'll talk a little bit more about that towards the end of this chapter. But for right now, the biggest thing to remember is the DNA. DNA is the most stable, and it is our blueprint, okay? So DNA is much more stable than RNA, and that's important because that is what we pass along to our offspring. If it was not stable and it was easily degraded, that introduces mutations and other pieces of potentially bad information that can flow from one blueprint to another. So if you're making a blueprint and that blueprint has a flaw where you forget to include the electrical panel or you put an elevator where there should be the plumbing or something like this, that can be an issue, okay? So this blueprint that we have that's more stable, DNA encodes all of our genes, okay? And these genes are what are passed along, right? And now these genes basically tell our offspring, have this color eye, make this kind of protein, do these different things. So genes are usually made into their components like proteins. Not all genes encode for a protein, but they can encode for other things, or they can regulate other pieces that we have of our DNA to open up the DNA or to close the DNA to make more or less of something. So again, genes go on to make RNA, to make protein, for example, in our central dogma. Okay. Now the thing is that mutations in our DNA can lead to diseases, to issues, things like this. So for example, a couple that your book touches on is something called sickle cell anemia. So sickle cell anemia is a mutation in the gene that codes for the protein hemoglobin. And when you have this mutation in your DNA, it leads to a difference in the structure of the protein. And if you remember from chapter 16, structure dictates function. So if you have the incorrect structure, you can have the incorrect function. It's the same way with genes, right? So if you have a mutation in your DNA, it can lead you potentially to the wrong or a mutation in the RNA, which can lead you to a different translation of the protein, which can therefore lead to defective in some way, shape, or form protein or resultant downstream issues. So again, the other example your book talks about are the BRCA1, BRCA2 genes, or BRCA genes. You may have heard of this before. People who have these mutations can get screened for them because they do increase your likelihood of experiencing breast cancer. So as you can see on the slides here to your left, right, we have DNA to DNA polymerase, RNA polymerase, and the ribosome as our translator. So again, it's this whole process of the central dogma, but there are pieces and other proteins and enzymes and things like this associated with making those DNA to RNA to protein types of things. So we are going to talk about the difference down at the biochemical structural level between DNA and RNA because they're really not all that different, which is really great. So if we take a look at these nucleotide structures, what we're going to look at, the difference between DNA and RNA is that difference in the D versus the R. The N and the A, actually, if you look, are exactly the same. So if you take a look at RNA, this stands for ribonucleic acid. So again, ribo stands for the fact that you have a ribose sugar. If you remember, we talked about ribose a little bit in the carbohydrate chapter, but we will draw the structure of it here in just a minute. So again, ribose is the monosaccharide, okay? Deoxyribonucleic acid is basically ribonucleic acid, but the only difference is that it is deoxy. Deoxy means without oxygen. So the big difference is that in RNA and DNA, it's a ribose sugar, but DNA is a ribose without an oxygen present on it. So both of them have a monosaccharide sugar, and if you notice that monosaccharide or the ribose that is sitting there, it is in a 5-membered ring. It is in the furanose form that we have for ribose. Okay? In addition to the furanose ring that is a ribose, the other pieces that you have on it are you have a nitrogenous base, I'll just abbreviate NB, and then you also have a phosphate group similar to what we saw with the lipid chapters. So again, this is the last major macromolecule we will discuss in this course, but again, these nucleic acids are physically bound together with covalent linkages, unlike lipids. So it's similar to carbohydrates when we make those disaccharide linkages, similar to proteins when we make the peptide bonds between those individual amino acids. So again, you have a ribose sugar in the furanose form, you have a phosphate, what we call backbone, and then you have the individual nucleic acids or nitrogenous bases that you have as you go through here. So again, deoxyribonucleic acid is without oxygen. So we're specifically going to investigate and look at, more specifically, the bases are slightly different between DNA and RNA, meaning there's only one that's different. The phosphate groups are exactly the same. The only difference between DNA and RNA has to do with one of the carbons, specifically carbon number 2 here, which we'll draw more in depth in just a minute, being a hydrogen instead of an OH group, so without that oxygen that's there. So again, looking at the slides, we can see DNA deoxyribonucleic acid. That is the difference. It's either an OH on RNA or an H on DNA, which we'll see more in depth in just a minute. And again, they both have ribose, they both have the sugar backbone, and they both have nitrogenous bases. The difference is, though, in RNA, one of those bases is uracil or U, and in DNA it is T or thiamine. So let's take a look more structurally investigative at this furanose ring that we sit on for DNA versus RNA. So remember, these are both ribose, but it's either ribose or deoxyribose. So for DNA, okay, we have, again, our ribose sugar, which is in our furanose form, in our 5-membered ring, okay? Ribose, if you remember, is an aldose, and it is a pentose, so it's got 5 carbons. So that pentose, right, we have CH2OH, this is carbon number 5, carbon 4, carbon 3, carbon 2, and our anomeric carbon, carbon number 1. Now, in DNA and RNA, our anomeric carbon is always beta, meaning carbon 5 and the anomeric carbon, the OH groups are in the same direction. So our OH group here will be up, okay? Remember, beta linkages are stronger, and that's actually what we want in this case, because we want our DNA to, frankly, quote unquote, survive when we go from parent to offspring. So if we take a look, the rest of our species, OH, OH, and then on carbon 4 here, right, we've got that remaining H. So here, this is just by itself ribose. So this is the ribose sugar. Now, carbon 2, that is the difference. So if you notice carbon 2 here, C2 in DNA will just be a hydrogen. C2 in RNA will be that OH, so deoxy. So here, as we've drawn this as an OH, this is RNA. If we change this to just an H, now we have DNA. That is the difference between the sugars in DNA versus RNA. That's the difference. That's the only difference. So if you notice, they're both in the furanose form, they're both in the beta form, they both have OH groups. The only difference is carbon number 2 in DNA is just without the oxygen, so just an H, and in RNA, it's an OH, okay? Those are the only differences between the sugars that are in the nucleotides. So if we take a look, though, we have differences between a nucleotide and a nucleoside, and then the nucleic acid that we have. So very quickly, when we take the sugar, which in this case is the ribose, and now we're going to add on here to this particular OH group, this beta linkage here, we will actually change this to what we call a beta N glycosidic linkage. Okay, so if you remember back from our carbohydrates chapter, we had a beta linkage because these two are both in the same position, they're both going up, but it is an N because this OH group, when it attaches to the nitrogenous base, it will attach to a nitrogen. Now that nitrogen changes depending upon what base you have, and meaning there's still a nitrogen, but the structure on that nitrogen changes depending on what base you have. So again, it's the anomeric carbon, right? Carbon number 1, because this is an aldose, and it's always beta, so it's always going up, okay? And we're taking off the OH group and replacing that with a nitrogen with the rest of whatever ring may be on there. So we call it a beta N glycosidic linkage. So it's linking the sugar to the nitrogenous base to make now a nucleoside. So here are the nitrogenous bases that you have. So there are a total of 5. DNA has 4 nitrogenous bases. RNA also has 4. So you would think there would only be 4, but remember, there are slight differences between DNA and RNA, okay? The biggest differences in DNA, the 4 base pairs that we have are A, T, G, and C. In RNA, the base pairs that we have are A, U, G, and C. So if you notice, 3 out of the 4 are exactly the same, okay? The only difference is in DNA, T, which stands for thymine, is now replaced in RNA with uracil, okay? If we take a look at the rest of the nitrogenous bases that we have, if you notice here, as you're looking onto the slides, we have cytosine for C. If you notice, those are the same in DNA and RNA. We also have guanosine, okay, on the far right of the slide here, or guanine as the single base itself, guanosine with the sugar. Osine is when you have the nucleoside present. You have adenine, again, same as in DNA versus RNA. So if you take a look, all of these structures, right, you should be able to recognize these structures. I would not expect you to draw them, but you should know the difference between thymine, uracil, adenine, guanine, and you should know which ones are what we call purines and which ones are pyrimidines. So we'll talk about a mnemonic to remember that in just a second here. But if you notice, all of these, you have these nitrogens that are circled for every single base pair. That means that's the nitrogen that is attached to the ribose sugar in that beta N linkage. Remember, both of these are beta in that case. So if you take a look, the way to remember which ones are which, we have 2 different types of base pairs. We have purines and we have pyrimidines. Purines are A and G. So purines is a shorter name. Okay, so if you take a look at the structure of the purine, a shorter name means basically a larger structure. So you have 2 rings versus 1. If you forget which ones are purines, for example, the easy way to remember, if you remember this mnemonic, is remember that purines are pure purines as a G, gold. So if you remember as gold, or you can remember that on the periodic table, you can also remember pure silver, which is Ag. So A and G are your purines. That means the remaining bases are pyrimidines. So this is a longer name, so it's a smaller structure. So it's only one ring. And so you can remember these to say cut cytosine, uracil, and thymine, the pi pyrimidines, Py, to remember pyrimidines in there. So these are your nitrogenous bases. You can look at the structures in the slides as you see right here. But again, I will never ask you to draw these freehand, but you should be able to, given them, be able to recognize which one they are, and then you should be able to determine, is this a piece of DNA or RNA, by the presence or absence of either thymine or uracil to distinguish DNA versus RNA.